This video is going to talk about osmosis. And so, of course, in biology, we really like talking about water. We have all these special things about water. So, of course, we would have a special name and whole different section for talking about water going through our cell membranes. And of course, as you know, water is polar. We spent a whole time on that in the biochemistry unit and talked about how all of water's properties stem from the fact that it is a polar molecule. And of course, the inside of a membrane is hydrophobic, literally water fearing, okay? So how does water go across our cell membranes if the center of our cell membranes literally are afraid of water? And so to get around this, we have these protein channels called aquaporins. Think about what you hear there. Aqua, water, and porins, pores, channels, things like that. So aquaporins are going to be these protein channels, and they sort of look like this green blob here in this picture, that are just going to be channels, sort of tunnels for water molecules to go through. And so we have named this osmosis is going to be the diffusion of water through a selectively permeable membrane, much like our cell membranes. Okay, this over here on the right is just sort of a more scientific computer model of what an aquaporin actually looks like. So, again, because we love talking about water, we have these three different types of solutions that cells can be placed in, and it's all categorized on what is the relative concentration of water inside versus outside of the cell. So let's start all the way on the left here. A cell can be in a hypotonic solution. In a hypotonic solution, there is more water on the outside of the cell in the solution than there is on the inside of the cell. So there's less water, and I'll write less over here. So there's more water on the outside and less on the inside. Let's get some percentages there. Let's just say we're in pure distilled water, 100% water on the outside, and just for comparison's sake, 10% water on the inside. That's a little low, but there you go. And so as we know, things like to go from a high concentration to a low concentration. So in both of these cases, water will go from the area of high concentration, the outside, to the inside of the cell. And the cell, because of that, will swell. In the case of an animal cell, the cell will burst because we only have cell membranes. It can only expand so much, and it will burst. And we call this lyse, or it'll have lysed. And we know that lyse means to break, similar to lysosomes, those organelles that break things down in our cells. Plant cells, on the other hand, they will become turgid, sort of another word for stiff. And plant cells love being in hypotonic solutions. Think about it. You don't feed your plants Gatorade, you feed them pure water. They like being in a solution of pure water, and they won't burst because they have those protective cell walls. So in a hypotonic solution, there's more water on the outside than there is in the inside of the cell, and water goes into the cell. Old students of mine have said that in a hypotonic solution, a cell will swell like an O. I'm not sure if that will help you or not. In an isotonic solution, you have the same amount of water both inside and outside of the cell. So I'll just write equal and equal. So this cell and its solution are at equilibrium. And therefore, water does move. It just goes into and out of the cell at an equal rate. And so just for putting a number associated with it, let's say there's 70% water in the solution and 70% water in the cells. The sizes of the cells stay the same, everything stays the same, okay? And for animal cells like to be in an isotonic solution. That is normal for them. Plant cells, again, we said they like to be in a hypotonic solution. Last but not least, we have a hypertonic solution. In a hypertonic solution, there is less water on the outside than there is on the inside. So there's less in the solution and more inside the cell. And again, because water likes to go from an area of high concentration to a low concentration, water will move out of the cell from that area of high inside the cell to the area of low. And because water is moving out of the cell, it will shrivel. So an example of this, let's just say there was 70% water inside the cell and 30% water in the solution. Water wants to go to an area where it's less concentrated, so it will go out. So we will say that a cell is shriveled or plasmolized here. And of course, no cells like that. So again, sort of capping this, animals, for their animal cells, the cell membrane really will determine the ultimate size and shape. It can shrivel a lot, and it can burst if it gets too big. Plants, they have that cell wall. So you'll really notice the central vacuole changing a lot in size. And again, in plants, it cannot burst. But if you think about this, some significant amount of this type of science had to go into any medicine that goes through an IV. I mean, you're injecting a liquid straight into your bloodstream. And so you would probably like that your blood cells are in an isotonic solution. So in terms of concentrations of liquids going into your bloodstream, it can't be pure water, else your cells would burst. So even though this seems sort of like, when would you ever use this? If you were ever hooked up to an IV, you should be grateful.